guys, how you going? I wanted to read this because I found this really interesting. It's a book from obviously a .gov site. Effects of electromagnetic fields on organs and tissues. Introduction. A large body of literature exists on the response of tissues to electromagnetic fields, primarily in the extremely low frequency ELF and microwave frequency ranges. In general, the reported effects of radio frequency RF radiation on tissue and organ systems have been attributed to thermal interactions, although the existence of non-thermal effects at low field intensities is still a subject of active investigation. This chapter summarizes reported RF effects on major physiological systems and provides estimate of the threshold specific absorption rate, SARS, required to produce such effects. So that is specific absorption rates. Organ and tissue responses to ELF fields and attempts to characterize field thresholds are also summarized. The relevance of these findings to the possible association of health effects with exposure to RF fields from Gwen antennas is assessed. This is a .gov, mind you guys. And so they're talking about the Gwen antennas, the Gwen Towers, which I'm pretty sure is the mobile phone towers, correct me if I'm wrong. But those things have got all different shapes of things all over. Have you seen them? They actually look like speakers of some sort. Nervous system. The effects of radiation on nervous tissues have been a subject of active investigation since changes in animal behavior and nerve electrical properties were first reported in the Soviet Union during the 1950s and 1960s. RF radiation is reported to affect isolated nerve preparations, the central nervous system, brain chemistry and histology, and the blood-brain barrier. In studies with in vitro nerve preparations, changes have been observed in the firing rates of aplysia neurons and in the refractory period of isolated frog sciatic nerves exposed to 2.45 gigahertz microwaves at SAR values exceeding 5 watt per kilo. Those effects were very likely associated with heating of the nerve preparations, in that much higher SAR values have not been found to produce changes in the electrical properties of isolated nerves when the temperature was controlled. Studies on isolated heart preparations have provided evidence of bradycardia as a result of exposure to RF radiation at non-thermal power densities. Although some of the reported effects might have been artifacts caused by currents induced in the recording electrodes or by non-physiological conditions in the bathing medium. Several groups of investigators have reported that non-thermal levels of RF fields can alter Ca2 plus binding to the surfaces of nerve cells in isolated brain hemispheres and neuroblastoma cells cultured in vitro, reviewed by the World Health Organization, and in chapters 3 and 7 of this report. That phenomenon, however, is observed only when the RF field is amplitude modulated at extremely low frequencies. The maximum effect occurs at modulation frequency of 16 Hz. A similar effect has recently been reported in isolated frog hearts. The importance of changes in Ca2 plus binding on the functional properties of nerve cells has not been established, and there is no clear evidence that the reported effect of low-intensity amplitude modulated RF fields poses a substantial health risk. Okay guys, I had to edit this just to insert this to know what Ca2 plus is. It's calcium ions and they contribute to the physiology and biochemistry of organisms' cell. They play an important role in signal transduction pathways, where they act as a second messenger in neurotransmitter release from neurons, in contraction of all muscle cell types, and in fertilization. Many enzymes require calcium ions as a cofactor, including several of the coagulation factors. Extracellular calcium is also important for maintaining the potential difference across excitable cell membranes as well as proper bone formation. Plasma calcium levels in mammals are highly regulated, with bone acting as the major mineral storage site. Calcium ions, Ca2+, are released from bone into the bloodstream under controlled conditions. Calcium is transported through the bloodstream as dissolved ions or bound to proteins such as serum albumin. Parathyroid hormone, secreted by the parathyroid gland, regulates the resorption of Ca2 from bone, reabsorption in the kidney back into circulation, and increases in the activation of vitamin D3 to calcitriol. Calcitriol, the active form of vitamin D3, promotes absorption of calcium from the intestines and bones. Calcitonin, secreted from the parafollicular cells of the thyroid gland, also affects calcium levels by opposing parathyroid hormone. However, its physiological significance in humans is dubious. Now, this is just according to WikiLeaks. So now that we know what it is, let's go back. And carry on from where we were. Just keep that in mind. I just had to edit that in. So we understood what Ca2+ is. Results of in vivo studies, or vivo. Okay. Results of in vivo studies, both pulsed and continuous wave CF, RF fields on brain electrical activity, have indicated that transient effects can occur at SAR values exceeding 1 watt per kilo. Evidence has been presented that cholinergic activity of brain tissue is influenced by RF fields at SAR values as low as 0.45 watts per kilo. Is that right? That's what I'm assuming. Exposure to non-thermal RF radiation has been reported to influence the electroencephalograms, EEGs, of cats when the field was amplitude modulated at frequencies less than 25 Hz, which is the range of naturally occurring EEG frequencies. The rate of Ca2 plus exchange from cat brain tissue in vivo was observed to change in response to similar irradiation conditions. Comparable effects on Ca2 plus binding were not observed in rat cerebral tissue exposed to RF radiation, although the fields used were pulsed at EEG frequencies rather than amplitude modulated, as noted above. The physiological significance of small shifts in Ca2 plus binding at nerve cell surfaces is unclear. A wide variety of changes in brain chemistry and structure have been reported after exposure of animals to high-intensity RF fields. The changes including decreased concentrations of epinephrine, norepinephrine, dopamine, and 5-hydroxytryptamine changes in axonal structure. A decreased number of Purkinje cells. This is really trying my English, and that's my first language. Purkinje cells and structural alterations in the hypothalamic region. Those effects have generally been associated with RF intensities that produce substantial local heating in the brain. Extensive studies have been carried out to detect possible effects of RF radiation on the integrity of the blood-brain barrier. Although several reports have suggested that non-thermal RF radiation can influence the permeability of the blood-brain barrier, most of the experimental findings indicate that such effects result from local heating in the head in response to SAR values in excess of 2 watts per kilo. Changes in cerebral blood flow rate rather than direct changes in permeability to tracer molecules might also be incorrectly interpreted as changes in the properties of the blood-brain barrier. Effects of pulsed and sinusoidal ELF fields on the electrical activity of the nervous system have also been studied extensively. In general, only high-intensity sinusoidal electric fields or rapidly pulsed magnetic fields induce sufficient current density in tissue around 0.1 to 1.0 AM2 or higher to alter neuronal excitability and synaptic transmission or to produce neuromuscular stimulation. Somewhat lower thresholds have been observed for the induction of visual phosphenes discussed in the next section, and for influencing the electrical activity of a plesia pacemaker neurons when the frequency of the applied field matched the endogenous neuronal firing rate. Those effects, however, have been observed only with ELF frequencies and would not be expected to occur at the higher frequencies associated with Gwen transmitters. I'm turning into Elmer Fudd. Recent studies with human volunteers exposed to 60 Hz electric and magnetic fields with intensities comparable with those of high voltage power lines have shown no consistent effects on the EEG. Minor changes were observed in reaction time and heart rate, but the variations were within normal ranges. Visual system. Cataract development as a result of exposure of the eye to high-intensity RF radiation has been studied for more than 30 years. Extensive experiments have been carried out with rabbits to determine the dependence of Carcinogenesis on the frequency and intensity of RF fields and on exposure time. In general, the lowest thresholds for cataract induction have been observed with near-field exposure at 1 to 10 GHz and a power density greater than 100 MW/cm2, I have no idea what that means, applied for at least an hour is required. Ooh. Most of the evidence indicates that the mechanism of injury leading to lens opacity is thermal, and pulsed and CW microwave fields appear to have similar thresholds for producing cataracts. Multiple subthreshold exposures do not lead to cataracts if the time between exposures is long enough to permit the tissue to return to its normal temperature. At frequencies where the wavelength of the RF field is not well matched to the dimensions of the eye, cataracts are not produced, even at extremely high power densities approaching the lethal levels. Although it is difficult to extrapolate results from laboratory animals to humans, the threshold power density required to produce cataracts is expected to be similar in rabbits and humans because of the structural similarities and comparable dimensions of the eyes in these species. So they're experimenting on animals as well. And to me, they've just admitted, even at extremely high power densities approaching the lethal levels, and how do they know what the lethal levels are? They'd have to have killed animals to have got there, to understand where the lethal level is. Dear God. guys. Okay. Even though I knew this, I'm still surprised by what I read sometimes, guys. Results of recent studies with monkeys have indicated that vascular leakage can increase at relatively low power densities of pulse 2.45 gigahertz radiation when the eye is penetrated with, pre-treated with timolol malate, which decreases intraocular pressure by reducing the production of aqueous humor. Yep, okay, if you understand that, let me know what it means. A power density as low as 1, and if you understand what this is, 1 MW/cm square centimetre, I'm assuming. But what does the MW stand for? I might have to go and look. Corresponding to an intraocular SAR of 0.26 watt per kilo has been observed to produce the effect. Those findings might have implications for RF ocular damage in humans being treated with timolol malate for glaucoma. However, the threshold power density at 2.45 gigahertz is still well in excess of the intensity of the RF fields produced by Gwen antenna in areas accessible to the general public. A visual phenomenon associated with exposure to ELF fields that has been studied for nearly a century is the induction of a flickering illumination known as phosphenes. Time-varying magnetic fields with either pulsed or sinusoidal waveforms and frequencies below 100 Hz have been shown to produce phosphenes when the time rate of change of the field exceeds 1.3 T per second, 0.30. Okay, if you understand what that means, also put that below. The frequency most likely to produce magnetosphosphenes with sinusoidal fields is 20 Hz. The threshold flux density for eliciting the visual effect is 8 MT. A similar frequency dependence has been observed for electrophosphenes produced by placing electrodes in contact with the forehead near the eyes. The locus of the effect is in the retina, and available evidence suggests that induced currents in the retina elicit visual responses similar to those resulting from photic stimulation. Changes in visually evoked potential, VEP, have also been reported in response to ELF magnetic fields with flux densities that are 5 to 10 times greater than those required to produce phosphenes. Because phosphenes and VEP alterations are observed only with fields below 100 Hz, such phenomena are not expected to occur in response to the low frequency fields associated with Gwen antenna. The endocrine system. Many studies with rodents and monkeys, oh no, not monkeys, have demonstrated that exposure to thermogenic levels of RF radiation produces endocrine alterations, the most consistent change being an increase in plasma corticosterone. SAR values in excess of 3 watts per kilo produce an increase in plasma corticosterone in rats that depends on secretion of corticotropic hormones by the pituitary. Decreased thyroid hormone levels have also been observed in response to thermogenic levels of RF radiation, and this response has been associated with an inhibition of thyrotropin secretion by the pituitary. In general, the alteration of hormone concentrations is reversible after termination of the RF exposure. Those findings indicate that RF heating alters the complex interactions of the hypothalamic, pituitary, adrenal, and thyroid systems that are important in the maintenance of homeostasis. It It is noteworthy that a 2-year exposure of rats to non-thermogenic pulsed 2.45 GHz microwaves did not produce detectable endocrine alterations. Studies on the possible endocrine effects of ELF, electric and magnetic fields, have yielded inconsistent results. Increases, decreases, and no change in plasma steroid hormones have been reported. Results of studies with dogs and rats, Results of studies with dogs and rats suggest that the threshold of 60 Hz electric field required to produce changes in blood concentrations of corticosterone or testosterone in excesses of 10 kV per m, whatever that is. Results of experiments with monkeys exposed to 60 Hz electrical and magnetic fields at intensities typical of those in the vicinity of high voltage transmission lines indicated that a decrease in neurotransmitter concentration occurs during chronic exposure. However, there were no other observation of behavioural or physiological changes in the exposed animals. This is .gov, and they are experimenting on animals. And us, I'm sure. The most widely studied effect of ELF fields on the endocrine system is an apparent depression in the nocturnal rise of pineal melatonin. Changes in pineal melatonin have reportedly occurred after 2 to 3 weeks of exposure to electric fields with intensities exceeding 1.7 kilovolts per meter in air. The effect is reversible with a return of nocturnal pineal melatonin to control values within 3D of termination of exposure. 3 what? 3 days? 3 decades? Oh. A similar effect on pineal melatonin has been observed after exposure of rodents to a 0.05 MT static magnetic field that was continuously switched on and off in 5-minute cycles for 1 hour beginning 3.5 hours after the onset of darkness. Interest in this phenomenon has centered around the effects of melatonin on cell proliferation and its possible carcinogenetic effects. A major problem in the interpretation of the results of studies is lack of quantitative information on the threshold fields required to alter melatonin concentration. It is also unclear whether ELF fields directly alter pinealocyte functions or whether the reported alteration in pineal melatonin production is secondary to effects of the fields on the nervous system. Further studies are needed to assess the possible influence of field-induced changes in pineal melatonin on physiological regulation and the risk of endocrine-dependent cancers. Endocrine-dependent cancers. Sorry, I've been calling it endocrine. Oh, dear me. It is not now possible to extrapolate the available information obtained with 60 Hz fields or intermittent DC magnetic fields to the possible effects of fields with higher frequencies. Okay, immune system. Effects of RF field exposure on cellular components of the immune system have been reported with both in vitro and in vivo test systems. Oh, it is vivo. Okay. Lymphoblast transformation and changes in responsiveness to mutagens have been reported, although the effects observed in different laboratories, depending on how you say it, I might just call them labs for now, have been quite variable. It appears from available information that the threshold SAR for altering lymphocyte responses to mutagens exceeds 4 watt per kilo, and both pulsed and CW microwaves. Thermogenic levels of exposure have been found to decrease natural killer cell activity and to activate macrophages. The changes observed in components of the immune system at RF power densities that produce tissue heating are consistent with the expected effects of increased release of steroid hormones into the circulation. In one study involving a 2-year exposure of rats to non-thermal level of pulsed 2.45 gigahertz microwaves, SAR 0.4 watt per kilo, no significant irreversible changes were found in the concentrations of lymphocytes or their responses to mitogen stimulation. Numerous studies have been carried out to determine the effects of ELF, electric and magnetic fields, on components of the immune system. In general, sinusoidal ELF fields have been found to have no significant effects on immune competence after in vivo exposure of lab animals. However, reduced mitogen responses and decreased target cell toxicity have been reported for lymphocytes exposed in vitro to pulsed magnetic fields or 60 Hz amplitude modulated RF fields. These effects might have resulted from the relatively high current densities induced in the cell suspensions. In a study involving 60 Hz electric and magnetic fields with a sinusoidal waveform and intensities comparable with those of the fields near high voltage power lines, no effects were observed in the immunologic functions of peripheral human and canine lymphocytes obtained from donors that either were normal or had been challenged with specific antigens. Hematologic and cardiovascular systems. Several studies have been performed to assess the effects of both thermogenic and non-thermogenic levels of RF radiation on blood chemistry and blood cell counts. Most were conducted with 2.45 gigahertz microwaves, and the results indicate that power densities producing an average whole body SAR of less than 2.5 watts per kilo do not produce significant alterations in hematologic indexes. In one chronic exposure study with rabbits subjected to 2.45 gigahertz microwaves 23 hours a day for 180 days, I'm assuming that's what it is, a small decrease was observed in eosinophil count, serum albumin concentration, and calcium concentration. None of the other 38 blood characteristics measured in the body were found to change in response to chronic RF exposure. At high levels of RF exposures of mice, leading to rectal temperature increases of 2 to 4 degrees Celsius, decreased lymphocyte counts and increased neutrophil counts were observed. It was suggested that the release of adrenal steroids into the blood as a result of heat stress could have produced the changes in blood cell counts. Thermogenic levels of pulsed or CW microwaves might also affect the cellular composition and proliferative capacity of bone marrow cells. Both bradycardia and tachycardia have been observed in lab animals exposed to thermogenic levels of RF radiation with SARS values in excess of 2.5 watts per kilo. In general, the effects on cardiac dynamics were transient and consistent with effects expected from body heating. As reviewed by several authors, the threshold ELF fields for producing significant cardiovascular and hematologic alterations are high. For example, cardiovascular indexes were not affected by exposure to 60 Hz, 100 kVm fields. Acute human exposures to ELF electric fields up to 200 kVm and magnetic fields up to 5 MT have also failed to show any consistent hematologic or cardiovascular effects. Animal carcinogenesis. A few studies have investigated the carcinogenic potential of microwave radiation in whole animals. Male Swiss albino mice were exposed to radar transmitter with a 9.27 gigahertz frequency modulated with 2 USEC pulses at a pulse repetition frequency of 500 per second for 59 weeks, 5 days per week, and 4.5 minutes per day. The power used 1 kilowatt per square metre caused a temperature rise of 3.3 degrees Celsius. There was no difference between exposed and controlled animals in a number of characteristics examined, including body weight, red cell and white cell counts, and body temperature. Testicular degeneration occurred in 23 of 57, 40% of treated animals, and in 3 of 37, 8.1% of the controls. That's a bit of a difference. Monocytic or lymphatic organ tumours or myeloid leukemia was seen in 21 of 60, 35% treated, and 4 of 40, 10% control animals. However, that increase was seen in the animals killed at 16. Oh, they're killing them. They are killing them. At 60 MO-IMO after cessation of treatment, but not at 19 MO. In addition, there has been considerable criticism of the experimental methods. Example, definition of leukosis as an increase in circulating leukocytes, which could have been due to infection, and statistical analysis. Thus, the study is of questionable use for supporting an increase in cancer risk. The incident power density was approximately 100 times greater than the highest relevant Gwen levels. Yeah, but they perfected it. Female RFM mice were exposed to 0.8 gigahertz microwave radiation for 2 hours a day, 5 days a week for 35 weeks at 430 WM. I still can't get it. Red and white cell counts, hemoglobin, hematocrit activity, body weight, and survival were measured, but no histopathologic examinations were carried out. The only statistically significant finding was an increase in body weight of animals older than 86 weeks in exposed mice over control animals. A University of Washington study on male Sprague-Dawley rats was designed to stimulate the maximum absorbed power, 0.4 watts per kilo, of 0.45 gigahertz radiation. The frequency was chosen as typical of mid-range radar system. Rats were exposed at 2.45 gigahertz because it yielded a ratio of wavelength to maximum body dimension similar to that of children exposed at 0.45 gigahertz. Benign pheochromocytoma of the adrenal medulla was the only lesion with a statistically significant increase in incidence. However, that incidence was not higher than seen in control rats of other colonies. The time for appearance of first tumour was also shorter in treated animals, 457 days, than in control animals, 540 days. When all malignant tumours observed at all sites are combined, there is a statistically significant increase in incidence in the exposed animals. That holds true for carcinomas, but not for sarcomas. An effect on the process of carcinogenesis has been reported in several studies that use injected tumour cells or animals treated with low doses of a known carcinogen. The effects of 2.45 gigahertz microwave radiation at 50 and 150 watts per square metre in a anechoic chamber was determined. Lung sarcoma cells were injected intravenously intravenously into bulb C mice, and the lung cancer colonies were counted after 1, 2, and 3 months of treatment. After 3 months, the numbers of lung nodules were 3.6 plus 2.2.7.7 plus 2.0, 6.1 plus 8, and 10.8 plus 2.1 in controlled animals. If you get that, please explain it to me. Chronically stressed animals. Animals exposed at 50 watts per square metre and animals exposed at 150 watts per square metre, respectively. The time to appearance of spontaneous breast tumours in 50% of C3H HEA mice decreased in animals treated with chronic stress. 50 watts per square metre and 150 watts per square metre. 255, 261 and 219 days respectively, compared with 322 in controls. The time of appearance of skin tumours induced by benzoapiprine was also shortened when irradiation for 1 or 3 months preceded carcinogen application, or when radiation and carcinogen were given at the same time. Stress closely duplicated the effect of 50 watt per square metre of radiation, and although results of 150 watt per square metre were significantly greater than those of stress, or 50 watts per square metre, thermal effects might be responsible for the results at 150 watts per square metre. The power density of 50 watts per square metre is approximately 7 times the low frequency power density at the Gwen site boundary. Negative and beneficial results of exposure have also been reported. The effects of continuous or pulse waves of 2.5 GHz, 10 watts per square metre, SAR 1.2 watt per kilogram, was studied in black C57-6 mice with B16 melanoma. No significant effects on tumour development or survival times were observed. Beneficial effects of induced hypothermia have been observed after treatment with microwave radiation. So for cancer, all of this doesn't work. Is that what they're saying? I have no idea. It seems that way. Okay, no significant effects on tumour development or survival times were observed. Beneficial effects of induced hypothermia have been observed after treatment with microwave radiation. Lung sarcoma cells injected into BALB-C mice demonstrated temporary regression after exposure to 2.45 GHz of radiation. After radiation exposure was stopped, tumour volumes increased and lung metastases exceeded those in untreated animals. Sarcoma cells were implanted on postpartum day 16 into CFW mice that had been irradiated in utero with 2.45 GHz microwaves, 35 watts per kilo, during days 11 to 14 of gestation. The mice were then subject to additional exposure to microwave radiation. Fetal exposure to radiation that increased the dam's colonic temperatures by an average of 2.2 degrees Celsius decreased tumour incidence 13% with 46% in controls. Both tumour-bearing and tumour-free animals that were irradiated as fetuses live longer on the average than controls. Enhanced immunocompetence related to increases in temperature was suggested as an explanation. In summary, several studies have provided some evidence of possible carcinogenic potential, and others have shown no effect. Consistent reproducible studies demonstrating a dose-response relation in animals are lacking, and the interpretation of several studies is complicated by thermally induced stress. All studies were conducted with electromagnetic fields larger than the relevant Gwen fields. Considerable interest has arisen concerning a possible role of electromagnetic fields as co-carcinogens or cancer promoters. Results of cellular studies that support such speculation are discussed in Chapter 7. Studies by McLean et al. Have begun to explore directly the possible role of 60 Hz magnetic fields as cancer promoters. In their studies, cancer in Sencar mice, known for their sensitivity to the promoting effects of 12-O tetradecanol 13-acetate, TPA, were initiated with, oh, there goes my door, were initiated with, oh my goodness, dimethylbenzanthracene, and then promoted with TPA, a 2-MT magnetic field, or a combination of TPA and a magnetic field. Tumour development was then observed for 20 weeks. There was no significant difference in tumour development between those promoted with TPA alone and those promoted with TPA and a magnetic field. However, recent results of another experiment by the same group of investigators suggest that a 60 Hz magnetic field acts as a co-promoter. Additional studies are required to substantiate those findings. Benashvili et al. Treated rats with nitrosamethylurea and then exposed them to either a 0.2 gig static or 50 Hz magnetic field for either 0.5 or 3 hours per day for up to 2 years. They found an increased incidence and number of mammary tumours in the groups exposed to the magnetic fields, with the AC field being more active than the static one. They also reported an increased tumour response in rats exposed to the 50 Hz field alone. Conclusions. Very few studies have been performed on the responses of organs and tissues to electromagnetic fields in the low frequency or ultra-high frequency ranges used by Gwen transmitters. But it's possible to conclude from the analysis presented here that effects of RF radiation are unlikely to occur at the power densities and absorbed energies associated with Gwen fields. Are they sure about that? Because they just said very few studies have been performed. And they're concluding from this one that there's no effects are unlikely. The possible existence of non-thermal effects such as the reported effects of low-intensity, amplitude-modulated RF fields on Ca2 plus binding in nerve tissue do not alter that conclusion, in that the waveforms and frequency spectra of these fields are different from those of the Gwen fields. Physiological effects of ELF fields with frequencies less than 1/500th those of Gwen fields are generally associated with high field intensities and large induced current densities in tissue. Some physiologic effects of ELF fields that might result from low induced current in tissue, such as alterations in pineal melatonin concentration, have not been shown to pose a direct risk to human health. In some studies on physiologic effects of ELF fields have yielded little evidence that exposure to the low frequency fields from Gwen antenna in areas of most likely public access would represent a health risk. Excuse me. And then it goes into references. What do you think, guys? Because I'm thinking the conclusion was very few studies have been performed on the responses of organs and tissues to electromagnetic fields in the low frequency or ultra high frequencies ranges used by Gwen transmitters. But from this one study, are they getting all this information that they say it doesn't affect anything? Okay, guys, what I've done is I've edited this over 2 days. What I wanted to bring to your attention was something that stuck out to me. A large body of literature exists on the response of tissues to electromagnetic fields, primarily in the extremely low frequency ELF and microwave frequency ranges. Primarily. It doesn't mean all of them, but primarily. Okay, now let's go to the conclusion again. Now this is in the extremely low frequency and microwave frequencies. And then we come to conclusions. Very few studies have been performed on the responses of organs and tissues to electromagnetic fields in the low frequency or ultra high frequency ranges used by Gwen transmitters. And then they just say, but it's possible to conclude from the analysis presented here that the effects of RF radiation are unlikely to occur at the power densities and absorbed energies. So are they saying, just from this one study, because very few studies have been performed, but lots of literature exists? That had me confused. Anyway, my other conclusions are they're experimenting on animals, obviously in labs, and doing all sorts of things to them, including and up to when they pass away. They kill them, obviously. And they'd be experimenting on us because if very few studies have been performed on the responses of organs and tissues to electromagnetic fields in the low frequency or ultra high frequency ranges used by Gwen transmitters, then we're being experimented on. And I believe they know exactly which frequencies affect us. So this article to me is trying to make it seem okay. It's okay, there's no damage. But without really anything to back that up. Because it does show things. And then they're trying to contradict. It sounds like a contradiction. But you tell me what you think. This is a .gov website, so they're not going to tell us everything. And if you understand any of the terminology in this, like what I was thinking was we have megabytes, gigabytes, and terabytes. And what I was thinking is, do they have the same in hertz? Do they have megahertz, gigahertz, and terahertz? And is that what the T stood for? If you know, let me know down below. I should have had a look, guys, but if you know, please let me know. And I'm learning from you just as much as you are learning from me, guys. I hope I'm teaching you something anyway, even just by bringing this to people's attention, even though I'm just reading it. I'm hoping that we're all learning something from me reading this. So, yeah. Okay, let me know what you think in the comments, guys, about this one. This was a bit, to me, the whole thing was a bit strange. It was like they were trying to make it seem like it was okay, but that they did come across things that were going on. Anyway, okay, guys, I'll talk to you very soon. I'm just going to do some work on the other subject, and I shall be back hopefully within a day or two. Okay, guys. Bye.